Hello guys and welcome back to the foodborne microbiology. This series is going to continue with the hygiene and sanitary aspects of food. This chapter is going to be very dense and very big, so I'm going to split it into two parts and it's necessary for you to understand it better. So let's begin with the first part, but before we start, make sure to subscribe to the channel and comment I subscribed. I will do my best to reach and reply to each and every one of you. Thank you. It costs nothing to you and it helps me a lot. Okay, so we begin with spoiling microorganisms. Then we will view the gram-positive and gram-negative bacteria, and then the viruses and prions. Spoiling microorganisms, well, they cause alteration, either organoleptic, chemical, or physical. Useful life of food is the time when that food is considered safe and it maintains its properties and nutrition values. Physical and chemical and microbiological alteration, as I said. Perishable, semi-perishable, and non-perishable food. This is the classification based on how fast the degradation of the food occurs. Most changes are in the surface of the food, surface area. As we talked about in the last video of the intrinsic methods, of one of those was the surface area, if you remember. Microorganisms, we have molds and yeasts, the gram-negative bacteria, the gram-positive bacteria, the sporulated bacilli. I have to mention that these are all spoiling microorganisms. The microorganisms we are going to view next are the pathogenic ones. So spoiling molds and yeasts, they tolerate less water activity and less pH than bacteria, if you remember. And they act mainly on the vegetables. Gram-negative bacteria, such as these two families, they affect the meat and the eggs. The gram-positive bacteria, they affect the meat, the milk, because we have lactic acid bacteria, and the salads as well, the greens, the veggies. Sporulated bacilli, like the Bacillus and the Clostridium, they the Bacillus anaerobic facultative and mesophilic. The Clostridium are obligate anaerobes and they have a lower reduction oxidation point. And they affect mainly the bread and the canned or preserved food, like these ones. The metabolism, well, the alteration of the carbs is because of the fermentation. The pectin is degraded and the galacturonic acid is created. Then as well amylolytic enzymes that break down the starch will act, as well as the exopolysaccharides. In terms of protein, we will see an attack on protein anhydrolysis, and the amino acid degradation will cause the putrefaction that will cause the smell. And then the green color will start to form because of the stool for hemoglobin. Did you find a meat like with a green color? Well, that's because of the oxyhemoglobin together with the sulfuric. And then fats. Hydrolysis, well, it will break down the triglycerides into glycerol and fatty acids, and those fatty acids are going to form ketones, and ketones do smell a lot. These were the spoiling microorganisms. Moving on, we have the pathogenic bacteria and viruses and prions. So let's start with the gram-negative pathogenic bacteria. We have the Vibrio. Vibrio are bacilli, motile, flagellate, and they are aquatic. They are facultative anaerobes and catalase and oxidase positive. This is a method we're going to view in the next videos to classify the bacteria if they are positive or negative to these two tests. These two are, well, the Vibrio is positive to both of them. And we have Vibrio cholerae, Vibrio parahemolyticus, and Vibrio vulnificus. Vibrio cholerae creates the gastroenteritis and the cholera. It has the choleric toxin, which has two parts, the A and the B. The B will attach to the membrane, to the receptor on the membrane, and the A part is going to enter the enterocyte, bind to a G protein, and the G protein is going to activate the cyclic AMP. It will phosphorylate proteins, and then it will open the channel membranes to get out the bicarbonate and the chlorine, and it will block as well the entry of sodium. This will create a gradient outside the enterocyte in the lumen, and the water is going to follow that gradient to reach the osmosis, and the diarrhea will start to occur. Then the Vibrio cholerae also has the ZOT, which is used for the relaxation of the tight junctions between the enterocytes, and the ACE, the cholera toxin, as well as adenosines, biofilms, and other things they use as virulence factors. The oral fecal transmission is the main one, and you need more than 10 elevated at 8 bacteria to contract the disease. The prophylaxis, if we can say that, is red rotation, antibiotics, hygiene, and cooking to prevent the Vibrio, mainly in seafood. Vibrio parahemolyticus does the gastroenteritis during the summer because it likes all of them. They prefer the hot water, the warm water. It has adenosines and enteric toxins, and it's not invasive. It's associated with seafood and fish consumption, all of them are associated with seafood mainly, and the treatment is with antibiotics. Vibrio vulnificus, on the other hand, is the worst of the mole because it creates gastroenteritis as well as wound infections and the sepsis. This is very dangerous. It has a polysaccharide capsule as a protection. It is invasive in the bloodstream to the erythrocytes, and it's a flesh-eating bacteria. Causes more deaths than any other Vibrio, and the treatment is with antibiotics. Then we have the Enterobacteria family. Those are bacilli, flagellate, peritritic. If you remember, peritritic means the flagella all around the bacteria. Ubiquitous, that means they can be found everywhere, and they are facultative anaerobes. In this case, they are catalase positive and oxidase negative. We start with Escherichia coli. General characteristics of the Enterobacteria, and it is a motile bacteria, and it has the property of lactose fermentation. It causes gastroenteritis, urinary tract infection, systemic infections, and meningitis, depending on the type of Escherichia coli, because we have several ones. Starting with the enterotoxigenic, which causes the traveler's diarrhea. There is no human-to-human transmission, and it requires a high inoculum. And then we have the enteropathogens, which causes the childhood diarrhea in countries that are still trying to develop in poor countries. There is no such thing as very high human-to-human transmission. Indeed, it's very low, and the inoculum is low as well. Then we have the enteroaggregative, which causes the childhood diarrhea, but in rich countries, and it's related to growth alteration because it creates a damage to the microbiota. Then we have the worst. the enterohemorrhagic, which causes the bleeding diarrhea. There is a human-to-human transmission, low inoculum, so it's very dangerous because of those two things. And it's also named as O157H7. This is the number for the flagella, and this is for the polysaccharides on the surface. This is the one who has the Shigella toxin. The Shigella toxin creates the hemolytic uremic syndrome. Basically, the Shigella toxin enters the enterocytes, and not only the enterocytes, and it will cause the death of the cell. It will actually damage a lot the cell, and it will destroy it. Then we have the enteroinvasive, which causes the watery diarrhea and the colon infection, as well as ulcerations and fever. And as last one, we have the diffusely adherent, and these are no invasive and no toxins, and it lowers the absorption. And this is very important because as lowering the absorption, they affect children for their growth, because they're not be able to actually get those nutrients they need for the development of their bodies. And so the affected ones are in the children and elderly, as well as immunosuppressed people. The prophylaxis is radiation and antibiotics, as well as hygiene and food cooking. Then we have Salmonella. This is very, very popular. General characteristics as an Enterobacteria, and it has an enterotoxin, a cytotoxin, an antiphagocytic protein, and a complex 4 protector. There are two types of Salmonella. Salmonellosis typhimurium is the one we get like for ingesting raw eggs, raw chicken, and it causes gastroenteritis, nausea, and vomiting. There is no human-to-human transmission, and the diagnosis is systemic, and there's a self-limited treatment. The other one is the most dangerous one, which is the Salmonellosis typhi, which causes the typhoid fever. There is human-to-human transmission. No animals are involved in this, and it settles in the gallbladder. It causes also gastroenteritis at intervals, and the treatment is with antibiotics. Then we have Shigella. Shigella from the toxin Shiga. Well, immotile bacilli and no lactose fermentation, no capsule protection, and the human is a reservoir for this bacteria. It causes the shigellosis. It's an adhesion to the M cells between the enterocytes, and as I said, it has the Shigella toxin indeed, and it causes the hemolytic uremic syndrome. Indeed, we will feel abdominal spasms, bleeding diarrhea, as well as in the enterohemorrhagic Escherichia coli, the same Shigella toxin, bleeding diarrhea, and in this case also fever. And then there is human-to-human transmission as well as sexual transmission. Yersinia is a short bacilli psychotroph, which means that it prefers 15 to 30 degrees, but it can also grow at 0. It doesn't ferment the lactose, it's a urease positive, and it's immotile at 37 degrees, but it's motile at 25, so that means that indeed this bacteria prefers the lower temperature. It attacks the M cells in the ileum and settles in the mesenteric lymph nodes. Indeed, it survives immunity by hiding in those lymph nodes. It causes watery diarrhea, bleeding mucosa, and fever, and withstands vacuum in foodstuffs for immediate consumption. Then we have the gram-positive foodborne pathogenic bacteria, which are the Staphylococcus. Staphylococcus are immotile coccus, no spores. They have a capsule and they are facultative anaerobes catalase positive. Indeed, the Aureus species are coagulase positive as well. They can fermentate the glucose and the mannitol. They can create biofilms and they resist desiccation and salinity. The diseases can be either suppurative with pus or toxin-caused, no suppurative. Transmission is human to human, human to food, or animal to food. Pathogenesis survives a lot in food, is resistant to gastric acid, remember that, enzymes, as well as temperature, and is a super antigenic. This means that it triggers a more pronounced immune response than the normal. And here we have to make a distinction between intoxication, which is poisoning, and toxin infection, which is the infection of the Staphylococcus, because it has toxin caused, that can lead to the gastroenteritis with the diarrhea and vomits. But if it is present as an toxic infection, we talk about enterocolitis, which is gastroenteritis and fever. The treatment is rehydration and maybe antibiotics, and the prophylaxis, isolation and culture of the bacteria in a laboratory to study the toxin. Bacillus spp, well, it's a bacilli, of course. Facultative anaerobes, they create endospores and they are catalase positive. Remember this, create endospores, because both are catalase positive, but here no spores, here endospores. The Cereus species grows in starch-rich or animal protein-rich foods, such as rice, for example. The rice is very common to have this Bacilluscereus. Indeed, the fried rice we left over for maybe a day or two, that's the perfect environment for this bacteria. It causes emetic gastroenteritis or diarrheic gastroenteritis. The emetic one is nausea and vomits, no diarrhea, and it's because of the intoxication, which is the poisoning. And the diarrheic gastroenteritis, it has diarrhea and nausea, no vomits, and it's toxic infection with the infection of the bacteria. The treatment is self-limited and lab isolation, and be careful with rice. Then we have Clostridium. Clostridium is a bacilli, creates endospores as well as bacillus. It's a strict anaerobe because it has fermentation, and it's a catalase and oxidase negative. Pathogenic capacities such as spores, speedy growth, and powerful enterotoxins. We have two types here, the Clostridium botulinum and the perfringens. The botulinum is a big bacilli. It's a peritritic. It has a peritritic flagella. Remember, all around the bacteria. Strict anaerobe, and it has no capsule. It causes the botulism using the botulinum toxins it has. The botulinum toxins block the muscle contraction. They block the acetylcholine from getting to the muscle tissues, and then the contraction cannot happen. We have two types of botulism. The foodborne botulism, which is in adults and is via poisoning, intoxication, and the infant botulism via toxic infection. Indeed, we ingest the Clostridium botulinum, the bacteria, ingestion of spores in honey, and this affects mainly the children because they are weak to that. The toxin is a monoprotease zinc dependent, And it blocks the acetylcholine signals in the neurons, as I said. It causes weakness. Because of that, we are not be able to actually contract well the muscles. Vomits, blurred vision, not only skeletal muscles but also muscles like the heart and the eyes in this case, and constipation, as well as the muscles of the gastrointestinal tract. We will not be able to impulse that steam through the intestine. The treatment is with the ventilatory support, antitoxins, gastric lavages, and it's a long recovery. Toxin detection in patients with a blood test is the diagnosis, and prophylaxis is don't eat swollen canned food, cook the food well, preach no honey to children. So refrigerate, cook, no honey to children, and don't eat the swollen canned food because they live in canned food. They like that anaerobic state. And then the Clostridium perfringens. It's an emotive bacilli, beta hemolytic, and histotoxic Clostridia can cause myonecrosis. This is very, very dangerous. It has a very considerable amount of toxins, and it indeed is classified with the toxins it has by toxinotypes, toxinotype A, B, C, D, or E, and they all contain lethal toxins. The cycle is, well, it starts with the spore, then germination, multiplication, ingestion of those germinated spores, sporulation inside our cells, and the enterotoxin is created. It can cause abnormal gastroenteritis or the necrotic enteritis, which is bleeding diarrhea, vomits, ulcers, and intestine perforations. Clinical diagnosis, radiation and antibiotics as a treatment. This is very, very, very dangerous. I didn't want to put a picture in here because it's very unpleasant. And the prophylaxis, hygiene, cooking at more than 60 degrees Celsius and freezing at minus 40. Click the like button below, share this video with your classmates, and download these diagrams I'm doing. They are on my website. They are free. See you on the next video. Bye.